Hello everyone. Science Spot is a podcast that shines a spotlight on the latest scientific technology from Japan. Your host is Ran from Science Talk. Today's topic is how a component found in wine, its astringency or bitterness, might hold the key to a new treatment for previously untreatable cancers. This research comes from a team at Tokyo Science University. And first, let's see the stage. In cancer treatment, antibody drugs, proteins that bind specifically to certain antigens to exert their therapeutic effects, have gained significant attention. We already have successful examples like trastuzumab, which targets HER2. However, traditional antibody drugs have primarily targeted antigens located on the cell surface. This is because antibodies are large, hydrophilic molecules that find it extremely difficult to pass through the cell's membrane. Imagine a highly skilled delivery service that can identify and neutralize harmful bad actors. Traditionally, these delivery agents could only operate on the exterior of buildings. This means cell surface. But many of the most dangerous bad actors are hiding deep inside the buildings, in the basement or inner rooms. Even if an antibody somehow got inside, it would get stuck in the building's internal mailroom. This means the endosomes, and able to reach its target. Previous attempts to get past these endosomes often used harsh methods that damaged the cell surface itself, leading to concern about cytotoxicity and the instability in the blood stream. This is where the newly developed polyphenol nanomachine comes in. This nanomachine is constructed using common components, polyphenol, specifically tannic acid, found in the wine and tea, and ferric ions. These form the metal polyphenol complex at the core of the nanomachine through coordination bonds. What makes this nanomachine is ingenious is its clever design. First, by combining a tannic acid with a stealthy polymer called polyethylene glycol, or PEG, they created a shell structure. This makes the nanomachine like invisible cloaks, allowing them to evade the body's immune system, remain stable in the bloodstream, and accumulate precisely at tumor tissues. These tiny nanomachines are only about 30 nanometers in diameter, that's thousands of times smaller than the weight of human hair. But the most remarkable features of this nanomachine is its escape ability from the endosomes. Once taken up by a cancer cell and trapped inside an endosome, the nanomachine senses the acidic environment within the endosome. This acidity triggers the dissociation of this complex structure, consuming a large number of protons. This process causes an increase in osmotic pressure inside the endosome, effectively making the endosome membrane burst from within and safely releasing the loaded antibody directly into the cell's cytoplasm, where its target awaits. Crucially, unlike previous techniques that used positively charged molecules, that nanomachine's charge is almost neutral, meaning minimal concerns about the cell cytotoxicity, and incredibly, it can be formed by simply mixing the antibody, polymer, and iron ions, making it simple to procedure for potential mass manufacturing. To test its efficacy, the researchers loaded the nanomachine with a therapeutic intracellular antibody, and which binds to an antigen expressed inside cancer cells. They then administered to a mouse model of triple negative breast cancer, a form of breast cancer notoriously difficult to treat with conventional antibody drugs. The results were truly impressive. Compared to untreated groups or groups given the antibody alone without the nanomachine, this new treatment successfully suppressed tumor growth to just 20% of the original size. This marks significant steps forward or for new therapies and against intractable cell cancers. The polyphenol nanomachine developed in this research is a groundbreaking technology that dramatically expands the applicability of antibody drugs from cell surface antigen to intracellular antigens. And this opens up new treatment options for intractable cancers that were previously unreachable. Furthermore, its high safety profile and the simple manufacturing process make it a highly promising candidate for future clinical application. I think this is a very great research for the treatment of cancer, but I think there are many ways to the escape of endosome, and I know the several methods for the breaking the membrane of the cell surface. But I think this technology is not special for the break the some membrane using the polyphenol. Polyphenol technology is very famous for using the break this membrane and escape the endosome. But the easy to make some the mixture or complex is very great point in this research. The manufacturing is very critical for the making some new drugs, but this research is the remarkable in the very the successful method by just mixing them. So this is very great. So that's all for today's Science Spot. This podcast is a broadcast daily on weekday morning in both Japanese and English. I hope today's discovery has changed how you view and the possibility of medical treatment. And I'd love for you to listen to the podcast and post your notes and thoughts with the hashtag ScienceBot. Thank you very much. See you next time.